Welcome to the NCOH monthly webinar. This month we will have presentations on COVID-19 and immunity. But before we continue, I would like to thank the NCOH communication team and the team from Squiffer for setting all this up. Now that we're all facing more stringent COVID-19 measures due to the increase in cases again, I'm sure we would all love to learn more on how our immune system is responding to cope with the virus. Why does it sometimes go into overdrive? And once we've had this infection, will our immunity be sufficient to protect us from the next episode? But before we continue, just a reminder that you can ask questions to us in the bar above your screen. You can also use the chat function. Please also remember to put the microphone on mute when you're not speaking. So let's start with the presentations. We start off with Professor Mihai Netea. Professor Mihai Netea is a physician and professor at the Radboud University Nijmegen, where he heads the Division of Experimental Medicine. He received the VD grant in 2005, a VICI grant in 2010, and European Research Council Consolidator grant in 2012. In 2016, he was awarded the Spinoza Prize. He is a member of the Academia Europaea since 2015 and of the Royal Netherlands Academy of Arts and Science in 2016. So we are very, very grateful that he could join us today. And he is now mostly known for his breakthrough research in the area of trained immunity. Professor Mihai Netea, the floor is yours.
Thank you very much for your kind introduction and for inviting me to share with you some of our data. And now I will share my screen, and I hope this will be seen by everyone. If not, please let me know. So in the next 10 minutes or so, I would like to discuss with you a little bit, very briefly, of course, because we do not have much time, regarding the innate immune responses in COVID-19 and how we can make them relevant for vaccination and how can we approach them from a vaccination point of view. And first of all, I would like to show you what is happening at the moment that SARS-CoV-2, the virus, invades the body. There are specific viral receptors, especially on epithelial cells, ACE2, in collaboration with TMPRSS2. They contribute to the entrance of the virus in the cells, and they release the viral genomic RNA. Thereafter, the virus uses the host cell to create new virus particles and then to spread further. In addition to that, of course, the host also tries to impede the multiplication of the virus and to fight the infection. And in an early stage, what is happening is actually that alveolar macrophages and activated T cells produce cytokines, pro-inflammatory mediators that activate cells, activate NK cells, activate thereafter an adaptive immune response to produce both antibodies and specific T cells against the virus, and they contribute to the elimination of the infection. Most of the time, fortunately, in the majority of the individuals, these processes are enough to eliminate the virus. However, in a small but significant percentage of the individuals, this process is not able to eliminate quickly the virus, and there is an increased inflammatory reaction with high viral loads in especially the lungs of the patients, which attract more immune cells at the site of the infection, including neutrophils, which are very pro-inflammatory. There is more and more activation and release of pro-inflammatory cytokines of the complement system, what we call cytokine storm. They determine a local reaction in the lung that can be deleterious. We have studied actually which are the entire components, what is the, let's say, the phenotype of this inflammatory dysregulation in the lung of patients with severe COVID-19. And this is a proteome assessment of the inflammatory mediators in individuals with a severe COVID-19 infection compared with those with a low, non-severe COVID-19, and on the right hand with red dots are the pro-inflammatory mediators increased in the ICU patients compared with the non-ICU patients. And the conclusion from this proteomic assessment is the following, and I will give you just the conclusion because of time's sake. Upon the invasion of the host by the SARS-CoV-2 virus, there is also local cell lysis, which releases what we call danger-associated molecular patterns in the circulation. They are, some of them, preformed cytokines such as interleukin-1 alpha or interleukin-8. Some of them are also stimulated by macrophages in the alveolar, by alveolar macrophage to release cytokines. These cytokines produce also chemokines, and they induce the accumulation of more cells at the site of the infection. As long as this process is constrained to locally to where the infections take place, that can have a beneficial role. However, if that spreads and becomes systemically, and if it becomes also inflammatory in the entire lung, this leads also to activation of different cascades, such as complement cascades, kallikrein, bradykinin cascade, which can lead to local angioedema in the lung, respiratory insufficiency, and unfortunately, in some patients, even death. In parallel with that, there are also other inflammatory and cytokine mediators, which unfortunately are downregulated by the viral infection, such as stem cell factor and interleukin-7 receptor, which in turn inhibit lymphopoiesis in the bone marrow of the patients. And this we see as the lymphopenia that is a characteristic of the more severe patients. How can we use this knowledge actually to increase the resistance to infection? We know thus that in individuals with low innate immune responses, this can lead to high viremia. And the high viremia, as I explained to you previously, can lead to hyperinflammation, a lot of symptoms, high severity, and sometimes even death. What we would like thus to do is to improve, to boost these innate immune responses in the beginning of NK cells, monocytes, macrophages in the lung, so the multiplication of the virus is impeded, that we have a low viral particle numbers locally, but also in circulation, which leads to low inflammation, low symptoms, and survival of the host. How can we do that? One of these approaches could be to use one of the very old vaccines that we all have used for almost 100 years, and that is Bacille Calmette-Guerin, which has been used for a long time in newborn children to protect them against tuberculosis. However, interestingly, what it has been observed when BCG was introduced in a lot of countries, and this is the first study which was published almost 100 years ago in Sweden, these studies have shown that not only tuberculosis is reduced by BCG, but also other types of infections, among which respiratory tract infections and neonatal sepsis in these children, decreasing the mortality by more than 50%. How is this happening? This is happening through reprogramming, epigenetic, metabolic, and functional reprogramming of myeloid cells and natural killer cells, which are components of the innate immune responses. And because of time, we do not have time to go into details into that, but what is happening is that transcriptional regulation is changed after the vaccination, allowing for an increased response of these innate immune cells by processes which take place in the nucleus at the level of the histones, in the nucleus of the myeloid and NK cells, allowing an increased host response. We wanted to assess whether we can use this as a host defense against severe respiratory tract infections and other types of infections, and we performed 2 years ago a randomized trial in which elderly individuals above the age of 65 years old were randomized to receive either a placebo or a BCG vaccination, and we followed them up to 1 year. And 2 months ago, we have identified that BCG vaccination has led in this 1 year actually to approximately 40% less infections in the vaccinated elderly compared with the placebo-treated group. And when we assessed which type of infections were less in the BCG vaccinated individuals, they were not intra-abdominal infections or urinary tract infections, but precisely as has been described in the children, BCG in the elderly protects especially against respiratory tract infections, including the viral infections, and these patients had 80% less infections compared with the placebo-treated individuals. All in all, we propose that improvement of innate immune responses, trained immunity as we called it, in the first phase of a pandemic can buy for us 1 or 2 years in which we can develop a specific vaccine, and that development of the specific vaccine can be used thereafter. Thus, currently there are several randomized trials with vaccine which can induce these processes. There are ongoing in the world more than 15 trials with BCG vaccination, 2 with oral polio vaccine, and 2 with measles-containing vaccines. With that, I would like to finish my presentation and thank, of course, all the colleagues who contributed to this work. Thank you very much.
Wow, thank you, Professor Mihai. We don't have any questions yet, so please a reminder to everyone that if you have any questions, please add them in the bar above the screen, and we'll post them to the presenters. Professor Mihai, I do have a question for you. It is really interesting and really complex. And my question to you is, how well is the increased immune response understood? And what are the key questions that you would still like to research? And what are the fundamental mechanisms underlying this?
So there are several important questions that still need to be understood. What are the initial components? Why are some individuals developing such severe form of the infection and others not? Also trying to explain the differences in the innate immune responses during COVID-19 infections between men and women, and also very importantly, what is different between children and adults, and especially elderly, who become very severely ill? These are crucial questions that we need to answer. In addition to that, also some of the pathways are not yet known, and we need especially to understand what are the biomarkers that could indicate which type of immunotherapy should we use in COVID-19 infections.
So this is a lot to think about. I'm sure we're going to get some questions later. Oh, we have one question, Professor. Can BCG vaccination in people who have already had BCG vaccination as a child have or show the same improved effect on innate immunity upon revaccination?
As far as we know, yes, because unfortunately what we have observed that this general boosting of the innate immune responses is present for probably 2 or 3 years, not more than that. And thereafter it fades out basically. So somebody who was vaccinated as child for BCG probably does not show these effects. We have also vaccinated individuals or revaccinated, as I have to say, who received previously a BCG vaccine. There are no side effects, and the response is more or less the same as the newly vaccinated individuals.
So that's very interesting. Have you been revaccinated in response to this outbreak?
Not yet, because we followed the advice actually that we should wait the results of the trials, and as one of the people doing the trial, I was not allowed to participate in the trial.
Very well. Well, we would love to hear more from you once the trials are completed. It's very promising and interesting. Thank you, and we may have more questions later for you. Thank you very much for your time.
Thank you.
Then next we go. Something went wrong to Associate Professor Berend Jan Bos. He is working at the Utrecht University at the veterinary faculty with a main focus on how do viruses enter the host cell and which receptor molecules do they hijack for binding, and how can we develop intervention strategies that effectively target the cell entry of viruses. Professor Berend Jan Bos, the floor is all yours.
My name is Berend Jan Bos indeed. I'm not an immunologist. I'm a molecular virologist specialized in coronaviruses. But within my group, we developed interest in isolation and profiling of antibodies, protective antibodies against coronaviruses. And in my presentation, I will discuss the role of antibodies in protection against COVID-19. I'll show a little bit of the work that we did on this, but I'll also zoom out and try to discuss with you what we know and also what we don't know in this on this topic. So if we go to the next slide. So the way our immune system actually is built, and you probably know that, is that upon infection with a virus, we will make antibodies against any viral antigen as being non-self antigens. But for coronaviruses, it's actually the spike protein and antibodies against the spike protein that can inhibit virus replication. As this is the key protein for virus entry into the host cell and also the main target, as you can see on the electron micrograph, on the viral surface, but also on the surface of infected cells. So these spike binding antibodies, they can exert several functions, antiviral functions. They can block the spike protein in its two functions, that is receptor binding, as well as the fusion of the viral and the cellular membrane. And that prevents the entry of the virus into the host cell. If you go to the next slide, yes. So due to the bivalency of antibodies, also antibodies targeting the spike can lead to virus aggregation. But in addition, these spike antibodies can also tag infected cells, but also viruses for destruction by immune effector cells. So there are also FC effector mediated functions. So because of all this, the spike protein is actually considered as the target for vaccine design. And actually many COVID-19 vaccine approaches are focusing on this. If you go to the next slide. So over the last couple of months, we have seen an explosion of articles coming out that describe the isolation of SARS coronavirus to neutralizing antibodies. And many research groups all around the world have actually used their advanced B-cell screening technology to mine the antibody repertoire of patients or immunized animals to look for neutralizing antibodies. And basically from all these studies, but also studies performed earlier on other coronaviruses, it's getting very clear that the most potent neutralizing antibodies are actually directed against the receptor binding domain that is colored here in different colors on the spike trimer. So this is basically the Achilles heel for the virus, as these antibodies can effectively neutralize infection, primarily by blocking, as you see on the right side, a receptor interaction. So on the next slide, we can, so early on in the pandemic, we also started to look at antibodies neutralizing SARS-CoV-2, and we used a collection of antibodies that we had developed with the group of Frank Grosveld at the Erasmus Medical Center that targeted the spike protein of the SARS coronavirus that emerged 70 years ago in 2003, and which is actually closely related to the SARS-CoV-2. So can you still see my slide? I can't see it anymore, but I trust you can. So I hope you can still see my slide. I can't see my own slides anymore. Okay, you can. Very good. So yes, I can see it again. So from this collection of SARS antibodies, we identified an antibody called 47011 that could also react with the spike protein of that SARS-CoV-2 virus, as you can see on the right side. So next slide. You can see that 47011 neutralized both viruses, so SARS-CoV-1 as well as SARS-CoV-2, And very recently, we also determined the cryo-EM structure of the spike protein in complex with the antibody. And you can see that on the right side that the antibody actually targets a very conserved part of the receptor binding domain, and explaining why it can bind to SARS-CoV-1 and 2 as well as other SARS-like viruses. You can also see that it binds away from the more variable region on the top that engages the receptor. So we also found that indeed this antibody does neutralize the virus, but by a receptor that is independent, by a mechanism that is independent of receptor binding blocking. So on the next slide, you can see the prophylactic efficacy of this antibody. Bart Haagmans and Body Rox from Erasmus Medical Center actually took this antibody and tested its prophylactic efficacy in a hamster model in comparison with convalescent plasma treatments. And as you can see on the different displays, the antibody on the left corner actually was able to reduce weight loss very much, as well as clinical signs on the right, and also macroscopically there were no lesions found in the lungs of the hamsters that were treated with the antibody. So this illustrates actually that administration of antibodies could be very useful in as an intervention strategy for COVID-19. So in the next slide, you can indeed see that some antibodies that have been discovered over the last couple of months are currently being developed as drugs for prophylactic as well as therapeutic use in humans, both for single antibodies as well as cocktails of antibodies, and they are at different stages of development. And the next slide. So there was an announcement 2 weeks ago that indeed these antibodies can do some good in humans. So Eli Lilly, one of the pharma companies that is currently developing such an antibody, announced that their antibody was indeed reducing the risk of or the need for hospitalization with 72%. So that is a considerable amount. And also recently, another company announced very good first clinical data on antibody treatments of patients. Next slide. So so as this virus is new to basically everyone, our immune system needs time to develop antibodies against this new virus. So one of the things is that, of course, our adaptive immune system takes time to develop antibodies, and on average this takes 7 to 14 days before we can detect the first antibodies, and it peaks around 14 days. But the level of serum neutralizing activity was in general quite low, and this may compromise also plasma treatments of patients. Also, neutralizing antibodies do correlate with the anti-RBD antibodies, confirming the profiling studies of the monoclonal antibodies I just mentioned. And neutralizing activity seems to correlate with age, but also with duration and severity of symptoms. But there are still many things known. If we go to the next point, for instance, how do the presence of antibodies impact the clinical course and severity of disease? If you go back, I didn't finish all the points. How can these levels of antibody, what kind of levels of antibodies do we need to protect against infectious disease? That is an important parameter we don't know. On the next level, if you click on the next. Okay. One of the other questions is indeed whether antibodies against the spike protein can even exacerbate COVID-19 through a process that is called antibody-dependent enhancement, ADE. So there are two mechanisms known by which this can occur. So ADE can occur via enhanced infection through an antibody-dependent manner through Fc reactions, interactions on immune cells on the top, which can lead to increased infection and increased virus production, as has been shown to play a role for flaviviruses such as dengue, as well as one coronavirus, FIPV, that infects macrophages. But ADE can also occur via enhanced immune activation, such as formation of immune complexes. So there is some concern that ADE is also a risk for SARS-2, and this is largely based on experiments that have been done with SARS-1. But I have to say that the reports on this are not consistent. So what it seems that so far there for COVID-19, there is no indication that ADE is a problem. And the best evidence perhaps comes from that the observation that treatment with convalescent serum from recovered patients did not result in adverse effects. But we have to be cautious, and of course this remains an area of further investigation. If we go to the next slide. So what can we learn from with respect to humoral immunity from the endemic human coronaviruses that are already circulating in the human population? And this picture actually comes from a very interesting study done by the group of Lia van der Hoek at AMC in Amsterdam, who looked at duration of immunity for the 4 known common cold coronaviruses that circulate in human, including HKU1, NL63, OC43, and 229E. And they determined reinfections in 10 individuals, actually, by looking at increase in antibody titers in serum samples that were collected over a long period of time. And the results are basically summarized here. What they observe is that there is an interval between infections with the same coronavirus that can be as short as 6 months. And clearly indicating that protective immunity against these common cold coronaviruses is short-lived. And of course, this is not very encouraging news if you consider, if you look forward to COVID-19, to SARS-CoV-2. But of course, we don't know how this translates to COVID-19, and also whether a low level of immunity that might be insufficient to prevent reinfection may still protect from severe disease. Next slide. So another unknown is actually how antibody responses to endemic viruses may or may not play a role in disease development of COVID-19. So here, this picture shows actually the level of sequence conservation between spike proteins of SARS-CoV-1, 2, and the endemic coronaviruses. And although the diversity is rather high, there are basically on the down part of the spike protein, there are some sites that are highly conserved and that might be targeted by broadly reactive antibodies. So on the right side, you see a graph from an interesting paper that reports on SARS-CoV-2 cross-reactive antibodies, and that were isolated from a convalescent SARS-CoV-1 donor. And basically, these cross-reactive antibodies not only reacted with SARS-CoV-1 and 2, but also with the spike proteins of endemic coronaviruses, showing that there are indeed cross-reactive antibodies. And the data from this paper suggests actually that cross-reactive antibodies are existing and that they reflect pre-existing B-cell responses against endemic coronaviruses that target conserved epitopes. But it remains to be seen how these cross-reactive antibodies are involved in disease, whether they are neutral or can protect or even play an enhancing role in disease. In the next slide, one of my last slides is that, is there any chance that that there will be antigenic variation? Of course, that can be that can compromise vaccine development and antibody-based therapies. So at this moment of time, the antigenic variation is very, very low because the virus actually went through a genetic bottleneck during the spillover event from animals to humans. But it will increase over time because of genetic variation. It's an RNA virus, but the question is whether antigenic variation will also play a role. In the next slides, you can see basically that the 229E endemic coronavirus is prone to antigenic drift. And this comes from a study of the group of Derini that shows that most of the sequence variation in the picture on the left is actually found in the receptor binding domain in the current 229E isolates, particularly in the receptor binding domain, and particularly also in the loops in the receptor binding domain on the right side, marked by the red bars, that engage the receptor. And these loops are targeted by the potent neutralizing antibodies. And this variation actually is accumulating over the years, over the last 50 years. So that suggests indeed that this common cold virus undergoes antigenic drift. And this indicates two things. It shows that antibodies indeed are involved in protection, and also secondly that antigenic drift may impose indeed antigenic liabilities for vaccines and antibody-based therapeutics that are currently being developed. But whether and how fast this antigenic drift will occur for SARS-2, again, that remains to be seen. Okay, and with that, I would like to thank, in the next slide, my colleagues at Utrecht University and also the collaborators from Erasmus Medical Center and Harvard Biomed, with whom we have a very nice collaboration over the last couple of years. I would acknowledge my funding sources. And in the next slide, I also would like to mention that last year we started NCOH PhD research program together with partners from Utrecht and Erasmus Medical Center, in which 4 PhD students started to work on actually antibody-based strategies against emerging viruses. So it's a very timely topic. So we have already enjoyed a number of very nice consortium meetings to discuss research progress. And hopefully the PhD students themselves will be able to present their work also during future scientific meetings within NCOH. With that, I would like to thank you for your attention.
Thank you, Bernd-Jan. Very, very interesting. And yes, we would love to hear more from your students on their research when they're ready to share. Although from the picture, it does seem they're working harder on Corona than COVID-19, to be very honest.
Yeah, we enjoyed the Corona beer at the last meeting, indeed. Yeah.
Very nice, very nice, very interesting. Although I find the message of the duration of protective immunity is something to think about for sure. That is not too encouraging. Yeah.
Yeah, it might be alarming, although I have to say, of course, that, well, there might be some level of immunity left on cellular or antibody immunity. And as long as that protects against severe disease, that would be already very good, of course.
Yes, yes, it would be very good. I have a couple of questions for you from the team. The first one is the short-lived immunity attributed to mutations in these coronaviruses that make them look like new to the immune system?
That's a very, very good question. So that's the question of antigenic drift and whether that plays a role indeed in how long the immunity lasts. So that's really an area that we should look into. And I think we can learn a lot there from the endemic coronaviruses that are around, that actually also came from animal reservoirs. Some of them, at least one of them, not that long ago, like 70 years ago, people date the introduction of that virus into the human population. So we can really learn from how these viruses actually are dealing with the immune responses, particularly antibody responses, whether antigenic drift plays a role there. I think it will be very predictive on how SARS-CoV-2 will behave in the future. So whether or not the short-lived immunity is partially caused by antigenic drift or not, that is the question. It also seems that for OC43 virus, another endemic coronavirus, there are different genetic clades that can be identified. And also there the question is how these relate to each other antigenically. So it's not just one virus that is around. There might be different viruses with different antigenic properties, and that also may play a role in the immunity that people are building up to one virus.
Right. Thank you. Another question is, could a repeated vaccination solve the problem of short-lived immunity?
Yeah, that's a good question. The question is actually, can we do better than the virus? Indeed, if it seems that natural infection doesn't provide you long-lasting immunity, maybe we can do better with a vaccine. That's a very good question. That is to be seen, and yeah, that will be very interesting to see how vaccination, how long the duration of immunity will be with a vaccine approach. Yeah.
Okay, very good. Well, I've got a couple of more questions, but I'm also conscious of time, so I propose we continue with the next speaker, and then we'll, in the plenary session, we'll pose the next questions for you. So thank you very much.
You're welcome.
So our next speaker is Rory de Vries, assistant professor at Erasmus Medical Center, Department of Virus Science. Rory is known to have pushed the boundary working with T cells. He showed that in a non-human primate model, measles vaccination protected macaques from disease caused by distinct but related animal viruses, due to the presence of cross-reactive T cells. Taking this one step further, he identified measles virus-specific T cells that recognize many other viruses within the same virus family, the paramyxoviridae. These cross-reactive T cells recognize epitopes of paramyxoviruses that are highly genetically conserved, owing to strong functional constraints that prevent mutations. This led him to start a postdoctoral project to develop an innovative universal influenza vaccine by genetically designing and optimizing viral protein to induce influenza virus-specific cross-reactive antibodies in T cells. But of course, today we're talking about COVID-19. So today we're learning about SARS-CoV-2 specific T cell responses in COVID-19 patients. Rory, the floor is all yours.
Thank you, Marta, also for the kind introduction and also for the chance to present my work here. And indeed, I am not a coronavirus virologist, but I'll do my best to show some nice data. I will share my screen. So I hope you all see my screen at the moment. So as Marta said, my name is Rory de Vries. I work at the Erasmus Medical Center in Rotterdam. And today I'll be presenting some data on SARS-CoV-2 specific T cells in COVID-19. And just like Berend-Jan did, I'll show some of our own data, but I will also be sharing a bird's eye view of what is known about T cells in COVID-19. So actually what was known from directly the beginning of the pandemic is that severe COVID-19 patients, and actually all COVID-19 patients, present with lymphopenia, and lymphopenia means low T cell counts. And this was seen both for CD4 and CD8 positive T cells. Well, later on, it was also found that this lymphopenia can be associated with disease severity. So what you see in these graphs here is that when you compare non-ICU patients with ICU patients often have a more severe lymphopenia. Well, what wasn't really clear from the start is that why these COVID-19 patients are actually lymphopenic, because we know the virus is not in blood, at least not, there's no viremia, so no virus that can be isolated from blood, although RNA has been reported. So it's not completely clear that why these patients are lymphopenic, although just like Professor Netea showed just now, interference with hematopoiesis might be a mechanism there. What was also known directly from the start of the pandemic is that COVID-19 patients almost directly after disease onset present with an increase in activated T cells. And that's shown here in this graph by presence of HLA-DR positive, CD38 positive T cells. And that was seen quite early. And that's actually a response that's seen in many viral diseases. So this is also seen in influenza, but also, for example, in Ebola. So that is the more general T cell response. Of course, the next question is, what do we know about these SARS-CoV-2 specific T cells? Well, at the moment, there is not much known. There are about 10 published papers out, so not the flurry of papers that Berend Jan showed on antibodies. However, there is a lot of preprints out, and I lost track of those. But in the end, if you take all the papers together, it is very clear, maybe even painfully clear, that we don't really understand what the role of specific T cells in disease really is. So if we go back to the original SARS-CoV epidemic in 2002 and 2003, it was shown that CD4 positive T cell responses are correlated to a positive outcome. So that means that CD4 and probably CD8 positive T cells are important for viral clearance, which also makes sense from a virology perspective. However, what's also known for COVID-19, and especially severe COVID-19, is that, and that's also what Professor Dea showed, is that these patients often have this immune hyperreactivity, where they present with high levels of all kinds of cytokines, for example, IL-6, IL-10, and IP-10. And that means that these T cells could also potentially play a role in immunopathology, maybe similar to what Berend Jan just showed now, the ADE-like mechanisms for antibodies. So altogether, that means that severe COVID-19 patients often present with lymphopenia, but also an immune hyperresponsiveness. So that means that there are definitely interactions between SARS-CoV-2 and the immune system that we need to fully understand. So with that, I'm showing you some of our own data, and this is a study in which we try to detect SARS-CoV-2 specific T cells in patients at the ICU suffering from ARDS, acute respiratory distress syndrome, caused by SARS-CoV-2. And I'm showing you data from 10 patients, and I actually compare those data to 10 age-matched healthy controls. And these healthy controls are blood samples that are taken pre-pandemic. So these are definitely controls never exposed to SARS-CoV-2. Although I say 10 versus 10, we have now expanded these cohorts quite extensively. So I think we are now talking about 20 versus 50 or so. So the first thing we observed is what was already known, and what I've shown you is that these ARDS patients, so severe COVID-19 patients, presented with a lymphopenia, as you can see here, in low CD3 positive T cell counts. We also observed an increased CD4 over CD8 ratio, which I haven't seen in other publications yet. So that means that these COVID-19 patients either have an expansion of CD4s or a depletion of CD8s going on. So if we then really look into the specificity of these T cells, what we did is we took PBMCs. As I said, we had healthy controls and COVID-19 patients, and we stimulate these with specific peptide pools. These peptide pools can be used to measure T cells in a specific manner, and the readout we use for that is flow cytometry. So that means you take PBMC from patients, you stimulate them with these peptide pools, and after 20 hours, you look for activation of T cells with flow cytometry. And in this case, we looked at upregulation of two surface markers, CD69 and CD137. Well, if we did that experiment, and what you can see here, in healthy controls, you hardly observe any responses after, in this case, peptide pool stimulation in comparison to our control, which is the MSO. And if you do the same experiment for COVID-19, you can see that these patients clearly have SARS-CoV-2 specific T cells. And if you then compare the groups, there was a clear significant difference. But however, the main readout we used was this so-called stimulation index. And that's actually a readout that's often used in the T cell field. And this is nothing different than a ratio between your activation of T cells after peptide stimulation over activation of T cells after vehicle stimulation. So just a ratio of your peptides over DMSO in this case. You end up with a stimulation index or a ratio, and if that is higher than 3, individuals are regarded responders. So that means they would have SARS-CoV-2 specific CD4 T cells in this case. And as you can see in our small cohort of 10 ICU patients, we detected SARS-CoV-2 specific CD4s in all 10. In healthy controls, we also had 1 responder, which I will come back to later on. So this entire experiment was done with a peptide pool that only covered the spike protein. We have done a similar experiment with a peptide pool that covers all other proteins but the spike protein. So, and you have to imagine that these are large peptide pools, so easily containing well over 200 peptides. And if we use this other peptide pool with covers all the other proteins, actually the result looked very similar. And if you just focus on this bottom right graph, you can see, again, all 10 COVID-19 patients had SARS-CoV-2 specific CD4s. And in this case, we detected responses in 2 out of 10 healthy controls. Well, because we use flow cytometry as a readout, it's quite easy to phenotype these T cells. And what we found is that the specific CD4 positive T cells that respond to peptide stimulation mainly had a central memory phenotype, and that's really a proliferative phenotype. Well, for sake of time, I will not go through the CD8 data, but we have done a very similar experiment for detection of CD8s. And what we found is that COVID-19 ARDS patients also have SARS-CoV-2 specific CD8 positive T cells, although the frequency was a bit lower, so we didn't detect it in 100% of the cases, more around 60 to 80% of the cases. And what we also found is that the phenotype of these CD8 T cells was different. It was more of an effector phenotype than a central memory phenotype. So having said that, I'll step away a little bit from our own data, and others have now also shown these SARS-CoV-2 specific T cells in different disease severities. And the next obvious question is, if we have these T cells, what are then the targets of these T cells? So what was done in this study, which is a study done in La Jolla with a group with which we actively collaborate, is they took PBMC from mild COVID-19 patients in this case and stimulated those with peptide pools covering all the different open reading frames. And they again calculated this stimulation index, which we had done as well, which I've showed you before. Well, if you look at the data, both in CD4s and in CD8s, they found that T cells mainly target the matrix nucleocapsid and spike protein. So those were really the prominent targets, both for CD4s and for CD8s. Well, in their study, they claim that this makes sense, and actually it does, because if you now look at in what abundance these proteins are produced, actually spike, matrix, and nucleocapsids are the proteins that are produced most abundantly. From a T cell biology perspective, it makes sense in that way that you would respond to these immunogens, because abundant production would lead to abundant peptides, would lead to a lot of peptide presentation to T cells. So that is their explanation of why S, M, and N are the most prominent targets. So I promised I would come back to this case of healthy control. So this is a small overview of the data that we had obtained. So we see CD4 and CD8 responses in COVID-19 patients, but I told you we also see responses in these healthy controls. In our case, this was around 20%. Well, we are a relatively small control cohort, but others did similar experiment. This is a study done in Germany where they found a response in healthy controls in 35%. And the study done in the US showed responses in healthy controls 9, 10, or 36%. And in all cases, these were pre-pandemic controls. So these were all controls that had never seen SARS-CoV-2. So of course, we asked ourselves the questions, and others as well, what is going on in these healthy controls? And obviously, there's one logical conclusion, and Berenjan alluded to this in antibodies as well. And it's similar for T cells. Are we looking at cross-reactivity? So are these actually control donors that were exposed to seasonal coronaviruses and therefore have T cells that can cross-react with the new SARS-CoV-2 virus? Well, there are two publications that now sort of show that, although it's not the best evidence yet. I will just briefly mention them. In this study, the authors made two different peptide pools, one covering the S1 part of the spike protein, the other one covering the S2 part of the spike protein, with the idea that S1 is less conserved and S2 is more conserved. And if you now look at the responses they found, especially in this middle graph, which they call reactive healthy donors, so those are the responders, they see that these didn't really react to the S1 part, but they did react to the S2 peptide pool. And from that, they conclude that these people recognize conserved regions, and therefore it's likely to speculate that the T cells are cross-reactive T cells. Well, then the group in the US did one additional experiment where they took PBMC from pre-pandemic donors and they stimulated them with peptides, SARS-CoV-2 peptides, and they generated these T cell clones and T cell lines. And what they observed is if they generate these SARS-CoV-2 specific T cell lines, these T cell lines could subsequently also recognize the different seasonal coronaviruses. So that is also on a small scale, some evidence for cross-reactivity. So it is very likely to assume that seasonal coronaviruses induce T cells that can cross-react with SARS-CoV-2. So the last question that I would like to answer in this webinar is about longevity. Berenjan also touched upon that with antibodies. And maybe with T cells, the situation is not so grim, but we don't know really yet. Of course, it hasn't been studied long-term for SARS-CoV-2 yet, but there is a recent study that was published in Nature, performed in Singapore, where PBMC were isolated from original SARS recovery. So these were people that recovered from SARS 17 years ago. They revisited the donors and took PBMC and stimulated these with peptide pools 17 years after the disease. And actually, in these SARS recoveries, they could still detect SARS-CoV specific T cells. So although we don't know yet for SARS-CoV-2, at least we do know that SARS-CoV specific T cells can be long-lived. So just to summarize, I've shown you that we can detect SARS-CoV-2 specific CD4s and CD8s in the blood of COVID-19 patients, and the predominant targets are spike matrix and nucleocapsid. I haven't shown you anything about cytokine productions, but we do know now that SARS-CoV-2 specific T cells in response to viral antigen predominantly produce dominant effector cytokines and Th1 cytokines. I've shown you the question about cross-reactivity, so it is likely that seasonal coronaviruses induce T cells that cross-react with SARS-CoV-2. I've shown you that SARS-CoV specific T cells can be long-lived. And what I haven't shown you, but what is an important conclusion as well, is that detection of SARS-CoV-2 specific T cells is an accurate measure of exposure. So there are now some publications out that show that SARS-CoV-2 recoveries or COVID-19 recoveries don't have antibodies. And actually in these individuals without antibodies, authors could always find SARS-CoV-2 specific T cells. So it could be a more accurate measure of exposure. And of course, I would like to leave you with the main question that we still have to ask ourselves and that we are studying at the moment. We need to understand what the role is of T cells in prevention of disease. And we really would like to understand what the role of these cross-reactive T cell is in prevention of disease, or maybe even exacerbation of disease. And with that, I'd like to thank the people involved at the Erasmus MC Virus Science Department, the doctors involved at the ICU department, and also our collaborators at LGI in La Jolla in the USA.
Thank you, Rory. Thank you very much for this uplifting presentation on the longevity of T cells. We have questions, but before I ask them, we also have limited time. I'm very curious if the presenters have questions regarding each other's presentations.
Yeah, I have one for Professor Netea. Is there already some preliminary data on the vaccination approach that you were talking about?
There are the preliminary data, or at least there is one study that we finished 3 months ago that I mentioned already on respiratory tract infections in the elderly. There are additional 15 ongoing studies. We don't have yet the results because fortunately during the summer the number of infections was very low. And there is one medRxiv paper published very recently, a couple of weeks ago, showing that revaccination of hospital personnel in United Arab Emirates, in a United Arab Emirates hospital, led to a very strong decrease in COVID-19 in the revaccinated healthcare personnel. But both the two published studies, ours and these medRxiv studies, are relatively small with 200-300 individuals. So the results of large phase 3 trials with thousands of individuals will become hopefully available at the end of the year or beginning of next year.
I'm very much looking forward to this these data, because I think also in terms of pandemic preparedness for the next outbreak of any virus, it will be very important to have these data, of course. Yes.
Indeed, the idea would be to use these in the beginning, so we have time to develop the specific vaccines which hopefully could give a longer term protection.
Professor Netea, you could expand a little bit on the effects on hematopoiesis, because I'm interested in lymphopenia and how that happens. So maybe you could share again what your view is on that.
So this is still very difficult, and I'm not a lymphocyte expert. So we have just observed when we performed a very broad proteomics assessment in the blood of very severe versus less severe individuals, that especially patients with COVID-19 have very low, with severe disease, have very low amounts, concentrations of some lymphopoietic factors, which, as I understand, are very important for lymphocyte development, such as stem cell factor and interleukin-7. And exactly how that is mediated, we do not know. But we hypothesize that at the level of the bone marrow, maybe also at the level of the thymus, although in the elderly, of course, the thymus is let's say, has devolved, or how to say it. So we believe that these tonic signals, which are necessary for the adaptive immune responses, are no longer present. It is very difficult exactly to know what should we do. I know that there were people proposing actually to use also interleukin-7 in the patients with severe COVID-19. Of course, we do not know yet exactly what those effects would be. There was one study done in sepsis, in bacterial sepsis with interleukin-7, which showed some promising results, but they were not clinical endpoints.
Thank you for that. As you can see, my background changed. It means we had a little glitch. We're back online. I have a few more questions before we go. Rory, does the presence of cross-reactive T cells actually protect against SARS-CoV-2 infection or disease?
I mean, that is what I've previously called the million-dollar question. We have no clue. However, what I would expect, just from a T cell biology perspective, is that if there is a level of protection, it's definitely only from disease and not from infection or transmission. So, my speculative answer would be maybe they protect from disease, but definitely not from infection and transmission.
Good, thank you. There are lots more questions, and what I'll do is I'll post them to the presenters, and we'll post them on the NCOH website in a summary of the presentation. So for now, I would really like to thank you, Berend-Jan, Rory, and Mihai, for your time and effort. Thank you very much for the sharp presentations. The next webinar is going to take place after the next annual scientific meeting, and the annual scientific meeting will be on the 29th of October. The topic will be COVID-19 from wildlife market to wild financial market. It's a longer discussion, and it's promising to be quite exciting. So I hope to all see you at our next annual scientific meeting. We'll send out an email notification as well, and there's more information on the NCOH. Thank you all to the viewers, and have a nice day.